right, so I hope that everyone is back from the break and you've had the chance to just stretch your legs a little bit. As I mentioned before, my name is Aleena and I'm part of the Ensembl outreach team, and we are responsible for delivering training courses, answering help desk tickets, and also managing social media pages. So I'll be speaking to you about the Ensembl Genome browser. And the way that the workshop is going to work is that we are going to have a presentation first, and I will be speaking to you about the background data, or how we produce a specific tool and process the data. And then we're going to go on to the Ensembl Genome browser, and then we will I'll take you through the website and show you where you can find all the different data and the tools. And after that, if we still have times, then we will have some exercises. So all of these exercises are available on our training page. So this is on training.ensembl.org. Don't worry, I'm going to share the link for this on the Zoom chat as well. So once you're on training.ensembl.org, then you can go to training material. This is on the blue navigation bar at the top. And from here, we can just click on the specific workshop. So this will be AQUA FAANG workshop. So that's the session for today. And within here, you will be able to find the PDF version to my presentation. We also have a living document. So it's up to you. We're not that many people present today for the session. So if you'd like, you can always ask the questions in the Zoom chat, or you can also use this living document, which is a shared Google document where you can ask questions, you can share screenshots. For example, if you're working through an exercise and you get stuck somewhere, you can share the screenshot here, and then I can get back to you on here as well. Similarly, if you cannot hear me properly, or if you think I need to slow down a little bit, or if you would like me to zoom in, then please do unmute yourself and let me know. Other than that, on this page, you will also find this section for demos and exercises. So every module that we're going to cover today and tomorrow has a specific tab. And within those tabs, there's some further sort of categories, if you will. And then we have a category for demo. So this is basically everything that I'm going to go through on my screen, but it's up to you whether you would like to follow me on the screen, or if you have another machine or another screen, then you can just follow these screenshots that are nicely annotated and labeled, so you can go through the demo on your own. And then underneath the demo, we also have this category for exercises. So this is where you'll be able to find session-specific exercises that you can work through. You'll find the text answer for these exercises below the questions as well, if you get stuck, and you can always work your way backwards. So I'll share the training page with you in the Zoom link, sorry, in the Zoom chat. And let's go back to the presentation. So are there any questions before I begin with my presentation? Any questions about the training page or any of the exercises? All right, so everything seems to be good. So let me just go into presentation mode. So the objectives for today's course are, we're going to talk about what Ensembl is, what type of data you can get from the Ensembl Genome browser. I'll show you how you can navigate the Ensembl Genome browser by going onto the website. And then we'll also talk about where you can go for help and where you can find documentation, as there is a lot of it available online on Ensembl, but also on ABI. So I'm going to start with a little bit of history. The first genome was sequenced by Fred Sanger and his team in 1977. And this was a small bacteriophage genome of about 5,000 bases. And this is what paved the way for sequencing of more complex organisms, including the draft for the human genome, which was published in early 2000s. It was a much larger genome of about 3 billion bases, and since then, many other organisms have been added to Ensembl. So the advances in sequencing technology has enabled the scientific community to sequence many different organisms. And now we have large consortia aiming to sequence all living organisms on Earth. But this has also enabled sequencing of many different individuals of a given species. So for example, different people of various ethnicities to assess the genetic diversity within the population and also their allele frequency. So Ensembl was started in 1999 when the human genome was in commission, as it was known that the automatic annotation would be essential. So Ensembl's goal was to automatically annotate the human genome and integrate this annotation with other biological data and make it all publicly available. And since Ensembl's launch in 2000, many other organisms have been added as well. So we have many other fish species available in Ensembl. So why do we need genome browsers like Ensembl? Genomes are big and they contain a lot of data, and we are producing them at a fairly massive rate. So if we look at the sequencing data, it can look a bit like this. So this is just a string of letters of genetic code, and this data is completely meaningless without annotation. The only way this data is going to mean something is if we work with this data and we process it. And this is what genome browsers do. So Ensembl is a type of genome browser that takes publicly available sequencing data from genomes. It's important to remember that we don't do any sequencing ourselves. We take publicly available sequencing data and then we make sense of this data. So we annotate genomes with genomic features like genes, variants, regulatory features like enhancers and promoters, and we also perform analysis on all of this data. Ensembl is also called added value resource, which means that it brings together information from a wide range of other databases in a single site and provides easy access to all of this data. We're not the only genome browser. We also have our sister page, which is called Ensembl Genomes, and this is the site that is dedicated to non-vertebrates. And then you might have also come across UCSC and also Genome Data Viewer from NCBI. So we have a lot of data in Ensembl. Genome assemblies are the primary data type that we import from publicly available databases. And then we annotate these genomes with features like with genes. These are called gene builds. And we have gene builds for over 300 vertebrate species. We have genetic variation data, not for every species, but for a very good representation of them. So for example, we have variation data for Atlantic salmon. We have comparative genomics that is calculated in-house, including genome alignments, gene trees, and homologs. And we also have regulatory build for a set of regulatory elements which are depicted from experimental data. Some of this is available for human and mouse, but we also have this regulation data coming up for many other species as well. Moving too fast. We also have some tools that help you access the Ensembl data. So we have Biomart, which is an online export data tool, and this allows you to export large amounts of Ensembl data without any programming knowledge. But we also have Variant Effect Predictor. So this is VEP, and this is a tool that you can use to annotate your own variants. We also allow you to display your own data in the browser, and we provide programmatic access to our data via APIs. Most importantly, Ensembl is completely open source, so it's completely free to use, and you can access our whole genome files that are stored in FTP and also GitHub. So I've already hinted that there are quite a few ways that you can access the Ensembl data. So you can access Ensembl's data one by one by using the browser, and that's where we're going to focus on today. But you can also access our data in bulk using tools such as Biomart and VEP, and we will speak about these tomorrow. We also have our APIs that you can use to access our data, and then we also have, you can also access our data directly via MySQL. And as I mentioned, we have our FTP site. So it's basically a public directory with whole genome flat files that are basically large files if you wanted to download genome-wide data. So Ensembl is dedicated to vertebrate genomes, and there are about 300 vertebrates, and the number is constantly growing. But you can also find non-vertebrate species. So you can find some non-vertebrate model organisms within the main Ensembl site. So this is fly, worm, and yeast genomes. And these are here because they're the most commonly used model organisms, but they're also there for some historical and comparative purposes as well. And as I mentioned previously, we also have Ensembl genomes, which is a site that is dedicated to non-vertebrates. And this is split by taxa. So you can find Ensembl bacteria, protists, fungi, metazoa, and plants within Ensembl genomes. And all of these sites basically follow the exact same logic as the main Ensembl site. They're just color-coded depending on the taxa. Ensembl is updated every 3 months as genomic data constantly changes. So our data is not static. Every 3 months, we release a new version, and behind the scenes, there is a lot of work. As new genome assemblies are input, we update our gene sets, we update our software, we run analysis on this new data, we update our variation and regulation data, and finally, we package all of this up to display it in the browser. The current Ensembl release is Ensembl 109, and for Ensembl genomes, it would be 56. And this was released in February 2023. So this means that if you come back to Ensembl in a few months' time, the data there might not be the same. Therefore, it's crucial to remember which version you're working with. And if you wanted to make your data consistent and you wanted to find the version, that you were using in the past, you can do so by accessing our archives. We store all of our versions in our archives, which are kept frozen in time for up to 5 years. And I will show you where you can find the archives when we move on to the demonstration. So let's talk about genome assemblies, which is the primary data type that you can find in Ensembl, and how the genome assemblies are being generated. So I'm showing you a very historical approach on the slide. And this is basically on how the first genome was assembled. The first human genome was assembled. I need to start by stressing that currently there is no sequencing technique which is able to read the entire chromosome. Therefore, the DNA first has to be fragmented, and the fragments are then sequenced. So back in the day, step 1 was to cut the DNA into chunks, and then the human DNA was cloned into bags. So these are bacterial artificial chromosomes, which were then grown in these bacterial cultures to amplify the number of DNA copies. And finally, the cloned DNA was sequenced from the bags using Sanger sequencing. Later on, there was an era of PCR amplification, and nowadays, you don't really need any amplification at all. So sequencing creates these so-called sequencing reads of varying lengths. And then depending on the technology, but in all cases, there is shortening of the genome, which is then matched up using overlaps to create this continuous sequence called contig. And here I have a bit of terminology that is associated with the genome assembly. So a set of sequenced back clones with overlaps plotted along the genome are called a tile path. So here we have the tile path, and each of these blue boxes is a sequence back clone, and you can see that there's some overlaps between them, but they are organized in order, so they're plotted along the genome. And then the back clones with trimmed overlaps are called contigs. So contigs here are shown as these different blocks of different shades of blue. And then a set of contigs stuck together with no gaps is called a scaffold. So here we have one scaffold, and then finally the scaffolds are assembled into a chromosome. So that was a bit of an introduction about the Ensembl genome browser and how the genome assemblies are created. We're now going to move on to the first demonstration for today. So I'm going to look at the Ensembl genome homepage, and we're going to look at how we can find information about the species and genome assemblies in Ensembl. So I'm going to jump out of my presentation, and we are going to go to www.ensembl.org. So I'll paste this in the chat as well. sorry, it should be www.ensembl.org. So this is what the Ensembl homepage looks like. We have a blue navigation bar at the top of the page. And here we have some shortcuts to our most popular tools. So BLAST, VEP, Biomod, but you can find a full list of tools by clicking on this tools link. Then we have a link to our FTP site for where it says downloads. And then you can find a lot of help and documentations in this help and docs link. You can also access our blog by clicking on this blog link over here. And the blog basically contains articles about any updates, any new features that have come about in Ensembl. We post updates there about the release as well. So any new data, any new genomes that are coming up with the new release. And we also, you can also find articles there about people behind Ensembl, so we can get to know the team a little bit more. Then on the right-hand side, we have this little overview of what Ensembl Genome Browser is. And then here, just underneath that text, we have the release number. So as I mentioned, that we are on Ensembl release 109, which was published in February 2023. And then we have some bullet points for the major updates that took place with this release. But you can find more release news by clicking on this link, which is going to take you to our blog post. We have our rapid release site. So this is a site that gets updated every 2 weeks with new assemblies and genes and protein annotation. You cannot find some further functionalities such as VEP or variation data over here, but we do support BLAST. There is some comparative genomics data available as well. But basically, this site you can access if you would like to look at any new genome assemblies that we have annotated. And then if I scroll further down, we have this tiny link at the end of the page on the right-hand side that says View in Archive Site. And if I click on this, it's going to give you a menu of all of the archives that are available. As I mentioned, our versions are kept frozen in time for up to 5 years. Over here, you'll also be able to find the link to the previous human genome assembly, which is GRCH37. But other than that, we can find the previous Ensembl releases like 108, 107, 106, and so on. I'm not going to go on to the previous versions, so I can just jump out of this menu by clicking on anywhere outside the menu or on this tick box over here. I'm going to scroll back up, and you'll notice that we have 2 search options available on this page. So we have one at the top right-hand side, just over here. So this option, if you were to use this, this is going to give you results across all species. However, if you would like your search to be species specific, then you can use this option in the center of the page. And from this drop-down menu over here, where it says all species, you can then select the species that you are interested in. So for example, if I select Atlantic salmon, just over here, and then if you were to type your gene name over here or your coordinates, a variant, a phenotype that you might be interested in, the search results are going to be salmon specific. Underneath this search option, we have this section for genomes. So you'll have your favorite genomes over here. By default, they're set to human, mouse, and zebrafish, but you can always edit these favorite genomes by clicking on this pen icon. However, to edit your favorite genomes, you will need to create an Ensembl account. It's completely free to create. You don't get any correspondence from us. And it also means that you get to save your jobs within Ensembl. To view the list of all species that we have in Ensembl, we can click on View Full List of All Species. So it's just over here in this Genomes section. And this takes me to a page that shows me a table of all of the genomes or all of the species that are present in Ensembl. So you can see that the table is quite huge, and you do have the option to filter this table. And you also have the option to export this table by clicking on this Excel icon. So for example, if we filtered this table by salmon, we can see that we have our Atlantic salmon over here. So in the first column, we have the common name, then we have the scientific name for it, we have the taxon ID for it, the current ensemble assembly that's being used, the accession number, and also the gene build method. And then we also have information on whether or not we have the variation data and the regulation data available for this. So Y indicates yes, whereas if this was blank, then it would mean that we don't have this data available. To find out further information about the Atlantic salmon species, I'm going to click on Atlantic salmon, and this is going to take me to the species specific page. So again, anything that you search over here in this search option is going to give you results that are going to be species specific. So in this case, they're going to be salmon specific. And then we have this section on genome assembly. So you can find more information about this genome assembly by clicking on more information and statistics. So I'm going to click on this in a moment, but you can also download the DNA sequence in the FASTA format. You can display your own data in Ensembl. And then we also show you the previous salmon assembly mentioned just over here. So this was present in Ensembl release 105, and then we link you out to that assembly. And then we have some example pages for a comparative genomics, regulation, gene annotation, and variation. I would like to find out more information about this genome assembly. So I'm going to click on more information and statistics. And then from here, we have some information on the assembly, on when it was submitted and by whom. We have information on some gene annotation process. And then on the right-hand side, we have these summary statistics that give us information on, again, on the assembly, the NSDC, and then we have the GCS accession code. We have the base pair length. We have the information on annotation provider and the method, when the gene build was started and when the gene build was last updated. And then we also have information on the gene counts. So we have information on the coding genes, the non-coding genes, pseudogenes, and gene transcripts. Okay, so this was everything that I wanted to show you about the species specific pages and how you can find out more information about a specific assembly that you might be interested in. I'm going to go back to the Ensembl homepage, and to do that, I can click on this Ensembl logo that is present on the left-hand side on the navigation bar at the top. And the next thing that I would like to show you is how you can look at a specific region of interest by searching for the coordinates. So I'm going to go back to my slides, and from here, I'm going to copy these coordinates, and these are coordinates for Atlantic salmon. And these are also mentioned on the training page. So if you would like to go through these, or if you'd like to go through the demo on your own, then these will be mentioned in Exploring Genomic Regions. And if you just expand this regions demo, you can find the coordinates just over here. So you can copy them from here. So I'm going to go back to the genome browser. And from here, I'm going to paste in my coordinates that I'm interested in the search option. And it's important that I specify which species I'm interested in, because genomic coordinates can be ambiguous. So we can find similar coordinates in some other species as well. So from this drop-down menu, I'm going to specify that I'm interested in Atlantic salmon. The way that these coordinates are formatted is that we will have the chromosome number first. So we're on chromosome 25. And then after the colon, we're going to have the start coordinate hyphen the end coordinate. Once we're happy, we can click on Go. So this takes me to the location tab. So this is the first tab that we're going to look at today, but I would like to mention here that Ensemble's data is organized in a tab system. So later on, I'll also show you the gene tab or the transcript tab, which will have information on genes or transcripts respectively. So this is the location tab. The first thing that I would like to bring your attention to is this question mark icon. So this question mark icon is available on most pages in Ensembl, and you can click this to find out more information about the features or the things that you see on these pages. So for example, here, the help page is going to show you the different images that you see, and these images will be labeled. So you can always go to it if you get stuck somewhere or you're not sure what the information given is. And then you will also find on the left-hand side some frequently asked questions, video tutorials, glossary, and also a link to contact help desk. I'm going to jump out of this page. So I'm just going to close this and go back to our location tab. Then on the left-hand side, we have these location-based displays that you can sort of explore. We will be going through them when we move on to the other modules, but basically these are just different displays over here that you can navigate to find out more information about this region of your interest. Then underneath the menu, we have some blue buttons that you can use to configure this page. So configure this page would allow you to add more data to this page or change the style of the different features that you're viewing on this page, as by default, we don't show you all of the data that is present, because then the page is going to take ages to load. We also have a section for custom tracks. You can also export your data. So if I just click on this export data on the left-hand side, you can export the data in a faster sequence or as bed format. We have some feature file formats over here, or as flat file, and so on. Then you also have the option to change the location. So you can change the coordinates here. 1 over here would denote forward strand, and then if it was -1, that would be reverse strand. You can also choose your flanking sequence, upstream or downstream. Then if I click on next, you can also choose the format of your output for your export. So this could be an HTML, text, or compressed text. I'm not going to export anything for now, so let's just jump out of this menu. Then you also have the option to share this page. So if you were to click on this button over here, it's going to generate a link for you, which is going to be version specific. So it's going to specify that this is from the February 2023 release. And then if you were to share this with your collaborators or colleagues, it's going to take them to exactly this release and this page. And then you also have the option to bookmark this page. So if we look at these images, we have 3 images over here. So our main view, so all of these images are basically of different resolution. So the next is more detailed than the last. So our first image that we're looking at is the chromosomal view, and our region of interest is indicated by this red box over here. So this is the region that we have specified by providing the coordinates. In the next image, which is a little bit more detailed, we have, this is called the 1 megabyte view. So this is the image that is centered around our region of interest. So region of interest is still shown as this red box. And then we can see the surrounding genomic region of 1 megabyte on each side. So the view allows you so if you were to click on this view and drag your mouse across, you can always mark this region, or you can click on jump to this region, which is then going to change the image below to match the region above that you have selected. I hope it makes sense, but do let me know if there are any questions. So the data here is arranged by tracks. So we have 1 track for Contig. So this is basically our reference genome. So this is this blue bar. And then we have a track for genes. So the genes are shown as boxes, and their names will be shown aligned. So their names will be shown below, aligned to the left edge of the box. And we'll also have these little arrows next to the gene names that are going to indicate the gene orientation. And then underneath that image, so if we look at our third image, this is our most detailed image on the bottom. So this is basically our entire red box. So this is only our region of interest. And you can see that there are a few more tracks that are available over here. So track is a genome browser specific term. It basically means specific type of data that is plotted along the genome. So we can add more tracks to this data, to this image. And to do that, we can click on configure this page on the left-hand side. So I'm going to click on configure this page. It's this blue button on the left-hand side. And this menu is going to show you the tracks that are already active. So all of these tracks are already present on our location tab, but you can add more tracks by either searching for a track name. So you can type for the track name over here in the search option, or you also have the option to go through this menu and select the tracks that you might be interested in. So we have tracks that are related to sequence and assembly, genes and transcripts, variation, regulation, comparative genomics, some repeat regions, and some information and decoration. So let's say that I would like to turn the track on for RNA-seq models. So this is on the left-hand side. I'm going to click on RNA-seq models. And this is basically going to now show me all of the RNA-seq models available, and I can choose the data as BAM files, gene models, or introns spanning reads. You can also click on show tutorial. So if I just click on this, it's going to basically tell you what all these different buttons and selections will do. I'm going to click on hide tutorial just for now, just to make the page appear a bit simpler. And let's say you can filter your RNA-seq models. So over here, we just have ENA. And from here, I'm going to select, let's say, heart. And I'm going to select BAM files as normal. I'm also going to select gene models as expanded without labels. So it's completely up to you how you would like your track style to appear on your image. We can also select, I'm also going to add another track. So let's say that I would like to add a track for repeats. So I'm going to click on repeat regions, also on the left-hand side. And then from here, I'm going to click on repeats teloist. So to turn this track on, I'm going to click on this box next to repeats, and this turns it on for me. If you would like to find out more information about what these different tracks mean, you can always click on this eye icon that's in the same line as the track. And this is basically going to tell you what these tracks titles mean. So basically repeats teloist is that tells us that repeat masker was used to find repeats and low complexity sequences using the teloist repeats library in RepBase. To apply these tracks to our image, we can click on this tick icon over here and save and close this menu. So I'm just going to wait for my data to load. All right, so my data has now loaded. And here we have information on our new tracks that we've added. So we have our heart RNA sequencing gene models over here. And then we also have these heart RNA sequencing alignment over here as well. And then for our repeats, we have these just over here. So all the repeats are plotted along the genome. If you've added a lot of different data tracks, you can always reset your configuration to go back to the default image. You can also reset track order. So for example, all of these tracks you can move them according to your priorities. So you can't change the order of these tracks, but if you would like to go back to the default track order, then you can just click on reset track order, and then this is going to take you to the default setting. You also have the option to scroll on this image. So you can select basically the action of your mouse. You can either scroll or you can draw on this image. So this is set to scroll for now, for example. So if now I click and drag my mouse across, it's going to scroll to a different region, and then it's going to update the page for you. Similarly, you can always draw on a region as well. So I've changed the action of my mouse to select a region, and now I can, if I click and drag on this image, I now have the option to mark this region or jump to this region. Okay, so we've now seen how you can find information about a specific genome assembly, and we've also looked at the location tab where you can look at a specific genomic coordinate of your interest and how you can add more data to that region. Now we're going to have some time for exercises. So I would like to bring your attention back to the training page. And the exercises that we're going to do is going to be for Ensembl species. So we have 2 available over here. And then we're also going to try out the exercises for exploring genomic regions. And we have 2 here as well. So let's say that I'm going to give about 15 to 20 minutes. And let's see how everyone gets on. And if there are any questions, please do let me know, as I would be happy to go through these exercises on the screen as well.